So the title is, how do we get back? It's what's coming up in the breeding pipeline. And to answer that, I'm sort of going to answer a different question, and that is there a genetic solution to crown rot? Can we actually solve crown rot just through genetics? Yes and no. So yes, we can. We do see genetic variability in a plant's response to crown rot. We see that out in commercial release varieties. And probably more so in breeding lines that we're working with. We do see a fair degree of variation in resistance to crown rot. The issue is though that there's no complete resistance. We talk about almost complete resistance in things like stripe rust, where you can actually pretty much stop the infection either through the hypersensitive response or through seedling resistance. But we don't get that with crown rot. Crown rot's a totally different beast. And resistance is only ever going to be partial, small steps here and there. So resistance is only ever going to be part of the solution. Management will always have a role in the control of this disease, so Steve will always have a job. Yeah. We've talked about these two terms a little bit, particularly in regards to nematodes, and resistance and tolerance also apply for crown rot. As I mentioned before, though, we don't get this complete resistance. We don't get this ability to stop the pathogen. It gets into the plant quite early on and is able to sit in the plant quite happily, and the plant can't just get rid of it. So we have what's called partial resistance. So either restricting the amount of biomass, or the fungal biomass in the plant, how much the pathogen can proliferate up and down the stem, and also delaying when the pathogen starts to proliferate. We use the amount of stem browning, or the amount of symptoms we see on the stem, as a proxy for measuring how much fungus is in the plant. We also seem to see a tolerance in crown rot, and it's the ability of a plant to yield despite being infected. Whether this is an implied tolerance, like what you might see with a resistant nematode that seems to be tolerant to the disease, it may well be the same with crown rot. Lines that are resistant to the pathogen may well suffer less yield loss. The way we look at this is through yield loss testing. So we've identified a number of QTLs, or in simple terms, genes or regions of the genome where resistance to crown rot has been affected. I see we as the plural. There's a range of groups working on this. We've got representatives from DAFQ, now USQ, University of Queensland, SARDI, and CSIRO, all working looking for resistance to crown rot. And so a few of these have also been confirmed in adult plant field tests. And we know that pyramiding these genes, or putting these genes together, or these QTLs together, will improve resistance. So from that you say, well, why haven't we solved crown rot resistance? Well, unfortunately the inheritance and resistance of crown rot is highly complex. There's lots of very small genes, each of a very small effect, adding together to create the resistance. And there are lots of epistatic genes, or background genes, which turn on and off the resistance. So we easily lose the resistance between crosses. Additionally, there's very little understanding of the mechanism of resistance. We don't know what these genes are doing or how they are actually causing the resistance in the sources that we use. So we may well be pyriding multiple QTLs of similar effect. And on top of that, we've also got what I like to refer to as broadly useful traits. So water use efficiency, drought tolerance, and also maturity all play a part in controlling crown rot. Adding to the difficulties, the sources of resistance that we use are largely highly unadapted. They're tall, very late, rust susceptible, poor yielding, low threshability, you name it, it doesn't have it. And this is an example. This is one of the best sources that we have is what's called 249. Take a wheat and then think of something that looks remotely the opposite to wheat, and this is what this is. It's got nothing that we want, but it's what we have to use as a source. And there's also difficulty in reliably phenotyping. The plant's response to crown rot is different seemingly every time you do it. It's not like rust where you can get a reliable phenotype in the glasshouse. There are strong environmental influences, and also yield testing at early generations is not possible. So this is why we need pre-breeding. So pre-breeding is a step back from the commercial breeding process, where we start to identify the sources, whether they be land races, synthetic wheats, and lines from international and local collections. We then have to improve these sources agronomically, while pyramiding or combining multiple sources of these pastoral resistance together so we can get a line that the breeding companies can then work with. So this is where these pre-breeding programs come in. And this is a GRDC supported program with the aim to deliver germplasm with enhanced levels of crown rot resistance and tolerance, ultimately to grain growers. I'll just quickly run through how we actually go about doing this. We actually manage our blocks to create high levels of disease. Instead of trying to rotate away, we actually rotate towards durum wheats, as well as adding artificial inoculum, just to guarantee a high level of infection. In these disease blocks, we can run large populations. These are breeding populations from crosses where we can select single plants that show the traits that we're after, and through multiple generations can select individual lines that combine not only the agronomic traits and the yield traits that we're after, but also the resistance and tolerance. These then go into early yield testing. Throughout these breeding programs, we have to target what commercially the breeding companies are looking for. Ultimately, we rely on the commercial breeding programs to take up these lines so they can make it through to commercial release. So they have to be competitive with all the other traits that the breeding companies are dealing with. This includes yield, rusts, and to a lesser extent, grain quality as well. From there, we start looking at tolerance testing. So this is grown as plus and minus inoculum trials, so plus and minus the disease. And this is where we can start pulling apart the crown rot effects. So what's actually, what the crown rot pathogen is doing to these breeding lines, pulling them apart from, say, terminal heat or terminal moisture stress, which out in a commercial field can actually sometimes be quite hard to differentiate. You might look at a crop and say there's 60–70% whiteheads in there, and there's a little bit of stem browning. I've saw some stem browning, therefore there's 70% whiteheads caused by crown rot. Often that may be the case, but also too in an environment where you see crown rot, you're also running into high levels of moisture stress and high heat stress at the end of the season, which also cause whiteheads. So we really need to be careful what we say is crown rot and what we say is just the environment. An example of one of these trials we can see on the left would be an inoculated plot, large numbers of whiteheads, fewer tillers per plant, compared to the right-hand side where you've got a small number of whiteheads, which is probably a little bit of background inoculum or just that terminal heat and terminal moisture. And this is where we can start saying that, well, this variety is certainly not susceptible, not tolerant or resistant to crown rot, let's get rid of it. And these are really useful trials. On top of that, we also have the ability to induce the crown rot symptoms through rainout shelters. And this is where we can actually roll on shelters over trials and guarantee that post-anthesis drought, guarantee the environment that you require to create crown rot symptoms. And this is a great facility, but it seems to create drought. In the 3 years that we've used them, we've stopped about 15 mils of rain altogether. So if I get rid of them, you guys will be happy. So yeah, great facilities though, and really make sure that we can make selections every single year, regardless of the environment. So I'll just really quickly run through some data from the last 3 or 4 years of trials. So these are yield loss trials conducted not only at PBI Narrabri but across the northern region from the other collaborators as well. And what we've actually identified is 40 lines with yield under crown rot equal to or exceeding, in many cases, sun top. So these are lines that not only are well adapted to our environment, but also have the resistance sources that we're after that can be used in commercial breeding programs. So these slides are in the book, so the writing is very small, I apologise. But what we can see is the red lines are the standards. So in the middle here we got sun top, and then moving down we got sun guard, wily dance down to the more susceptible Tavia, and this is yield under disease. So we have a number of lines on the left-hand side, significantly higher yielding than sun guard, sun top. You know, they have to change it, don't they? On top of that, so this is one side, this is the resistance, this is tolerance, sorry. On the other side of that is resistance. And this is where, particularly the DAFQ program, we've got Cassy sitting up in the back corner, has developed we've got a large range of lines with resistance higher than what previously was the best commercial source, Sunco, with a number of lines exceeding 249. And this is actually a really significant achievement, given that poor agronomic type that we were dealing with 249, we've now got sources of resistance that we can provide to breeding companies to include into their breeding programs that isn't going to have that drag, all that genetic drag, where you have to then improve the yield, improve the rust susceptibility, all those sorts of things. So these have high value in commercial breeding programs. So again, 249 down the bottom there, down the bottom here, and a number of lines not significantly worse than 249 in regards to resistance. So where to from here? So as Rob said, this is actually the end of a 5-year program and the start of another 5-year program. In here we're continuing to cross new sources together and improve the deployment of molecular markers for the selection of resistance and tolerance to make it easier for the commercial breeding programs to deploy these sources of resistance in their selections to make sure it makes it to growers in a timely manner. The other really key thing to start doing is to identifying the physiological mechanisms behind resistance and tolerance. What's actually driving the response? It seems like a very fundamental question, but it's something that we just don't have a great finger on. And tied up in that will be improved phenotyping methods. Thank you very much.